Hey, Triple Science. So we're going to talk today about this molecule that you have seen before and done some work with when you did organic chemistry. So it says be prepared to share 2 facts you already know and 2 questions you have. Now, obviously, that is impossible. But if you had any specific questions on this that you wanted to send me, that'd be amazing. So we're going to be talking about Gregor Mendel, and we are going to be talking about DNA and how we moved into understanding about inheritance. So we've already spoken about evolution. We've already spoken about theories of evolution and why Darwin's was important. And now we're going to talk about how that links in with what we know about genetics. Okay. So from Gregor Mendel to DNA. So you need to know what the structure of DNA, how the structure of DNA was discovered. That comes up in the first part of the biology. So this is something that you're going to go back and forth over several times. And we've already done some work on this. We're called the structure of DNA, describe Mendel's inheritance experiments, explain how that helped with the discovery of DNA and link it to organic chemistry module. And that's going to give you those grade 8s, grade 9s, if you can sort of link across the 2 subjects like that. So this is Mendel. Very, very, very important in the worlds of genetics. He's known as the father of modern genetics, and interestingly, he was a monk. So obviously, religious aspects, rather interesting because he believed in God and everything that God did, but he also was the first to notice and actually model experimentally inheritance. So he gave the term genetic crossing, and genetic crossing is where genes get mixed during reproduction. So we already know that we get 50% of our genetics from mum and 50% of our genetics from dad. It's exactly 50-50, but those genes get mixed, and you will see some characteristics from mum and some characteristics from dad. And he explained this. And the way he explained this was using plants, so pea plants like sweet peas that he had in his garden. And there is a terrible science joke because that's Jack and the Beanstalk. Got nothing to do with beans. So that doesn't really work. Enjoy. Okay, so there's some bite-sized link there that obviously doesn't work because this is a video, but you might be able to copy it. So this is another picture of Gregor Mendel, and he bred pea plants, and he found that when he crossed pink pea plants with white pea plants, all of the offspring that they had were pink. And so remember, don't forget that plants can reproduce sexually and asexually. So he would have taken some pollen from one and put it on the stamen of the other, and a tubule would have grown and seeds, etc. But all of the offspring for his pea plants were pink, not white. And so he thought, well, why is that? And then he bred those. So there's some pictures on the next few slides. So here we go. Pink and white pea plants. Bred them together, and all of the first generation offspring were pink. Okay. And just so you know, I'm going to use the term Punnett square. And a Punnett square is just literally a visual way to represent what genes you're crossing and how likely it is you are to inherit characteristics. So when you see a kind of square that looks a little bit like you're playing noughts and crosses, that's a Punnett square. We're going to have a go at doing a Punnett square, but I just want to talk you through the Punnett squares. So his first generation occurred, so they're the first lot of offspring. So he had his pink flower, his white flower, and he noticed that they all turned out pink, so therefore pink is a dominant colour. So he was the first person to use the words dominant and recessive. So the white flowers were recessive because we didn't see any of those genes. Now he was the first person to say, okay, half of your genes are from mum, half of your genes are from dad in the terms of these plants. And so therefore white must have been passed on but not seen. So in the first generation, the first lot of babies, if you like, the flowers were homozygous. Now homo means the same, zygous means of zygote, so of baby. So the first generation, the parent flowers were homozygous, which means that the pink flower, which was dominant, contained 2 genes that were dominant pink. So capital P, capital P. The white flowers were homozygous recessive, which means that all of the genes that the white flower contained were recessive, little p, little p. Now here is a task. Draw a Punnett square to show the first generation cross. What are the genotypes and phenotypes? If you think, I remember this, I can do this, I'll give it a go. Great. If not, follow my example on the next page. Okay, so I've got the homozygous white flower on the left, and that is little p. I've got the homozygous pink flower, capital P, and if you look at my Punnett square, pink is on the top and white is down the sides. Now what this means is all organisms have, or each gene, has two genes that it can pass on. So what this does is says, okay, well, if this one gets passed on and is crossed with this one, what are the possibilities that could be produced? And so this is a Punnett square, so it's a really good way to determine. Now, if this gene gets crossed with this gene, I don't know why the last one's not in. So if the big P gets crossed with that one and that one gets crossed with that one, we're going to have all little p's. Okay, I don't know why that one's not pink. That one is also pink. I don't know why that one is white. So 100% of the genotypes is big P, little p. 100% of the phenotypes are pink. That's what we see. And 100% are heterozygous. They've all got two different genes, and we can see which one's dominant because it's capital, but that's the colour we see. So it's 100% pink. So excuse that diagram. He then took two of those baby first generation plants and he crossed those. So that's big P, little p crossed with big P, little p. And he noticed that the second generation, so the grand flowers of the previous ones, he said that they were all pink apart from one white one. And he was like, how come one of them is white? And this is where he really realized that there must be two sets of genes, and you can see one but you can't see the other. Different forms of the same gene are called alleles. So we're looking at the gene for color here. There's white or pink, and they would be alleles. So second generation occurred because the offspring of the first generation were all heterozygous. They had one pink and one white. So draw a Punnett square to show the second generation cross. So we're going to look at the second generation cross. So we've got big P, little p, and all of the babies were pink. So let's go through. Big P, big P, big P, little p, big P, little p. But because they've both got these two little p's, that means that we have got a 25% chance, or 1 in 4 chance, of producing a white flower. And that's essentially the first time we understood that there were two sets of genetics at play for any one thing. So if pea plants have pink flowers, they were dominant, and white flowers are recessive. So a flower to be white, it must have 2 white alleles, recessive. If a flower is pink, it only has to have 1 pink allele, and that's because pink is the colour. So some characteristics are dominant and some are recessive. Dominant genes tend to beat recessive genes. You're more likely to see them expressed, and they will appear as a phenotype. So this is a good time to look in a mirror and see how many of these you've got. So generally in classrooms, more people will have brown eyes than blue eyes because brown eyes is dominant and blue eyes are recessive. So I've said before, in my family, everyone has blue eyes apart from me, which means that I've got 2 siblings, both got brown eyes. My parents have brown eyes. I'm the only blue-eyed one, which means that both of my parents must have been carrying recessive blue-eyed genes. Dimples are dominant. So if mum or dad has a dimple, it's more likely that you are going to show dimples. Freckles are dominant. Dangly earlobes are dominant as opposed to attached ones. Thin lips are recessive, and red hair is recessive. If you have a dominant trait, it's likely that you will carry a recessive one too, but you just will never see it or see it expressed. So just to put this into human context, while we've been talking about plants, humans have 23 pairs of chromosomes in every cell, and 2 chromosomes in each pair, or 2 genes in each pair of chromosome carry information for the same characteristics. So here's some chromosomes. So eye color, hair color, etc. Your body has 2 sets of information for all your characteristics, or 2 alleles, one from mum, one from dad. So you might have 1 set of information from eye color from mum, 1 from dad, but you will only show the dominant, or if you've got 2 sets of recessive. Just a little quick reminder on this, that remember, we get all of our genes in pairs. That's because the gametes, so on the left we've got the ovum or the egg cell, and on the right we have the sperm cell. These cells will undergo something called meiosis, which means that is the cell splitting. And so an egg cell has half of the genetic information for one human, so 23 single chromosomes. A sperm cell will have 23 chromosomes, or half the amount of chromosomes for an actual human. So during fertilization, so that's when the 2 nuclei of the sperm and the egg fuse together to make a brand new cell called a zygote. That has 23 pairs of chromosomes, half from mum, half of dad, brand new person.
random combinations of alleles and genes in those chromosomes. So you might get your mum's blue-eyed recessive gene, even though she's got brown eyes, etc. So some people say they look more like their mum, or they look more like their dad. That would be because those traits are dominant, or that trait has been carried by showing 2 recessive genes, and that's the reason why it's expressed. You don't look more like your mum because you get more genes from your mum. You get exactly 50-50 from mum and dad. So, and that moment occurs at the moment of conception and fertilization.
So here's another example. So this is one of the examples. So the mother has dimples, which is a dominant trait, but she's heterozygous, so she's got the dominant and the recessive one. The father doesn't have dimples and doesn't have a dominant gene, so he's homozygous recessive. So if they were to have babies, so you've got dad's possible genes on the top there, you've got mum's down the side, and so 2 out of the 4 have the capital D, so that means that there's a 2 in 4 chance of them having dimples inherited from the mother, which means that 50% of their children are likely to have dimples.
Sex determination, we know that all of the eggs that females produce will have an X chromosome, or the X gene, whereas sperm can either carry the X gene or the Y gene. So males are heterozygous and women are homozygous when it comes to the sex determination gene. Now, if you look at this Punnett square, male on top, lady on side, female on side, you can see that if that egg meets that sperm, then that's going to be a girl. If that egg meets the one on the far right sperm, then that's going to be a boy. And so there's always 50-50 chance of having a girl or a boy. I wanted to just include this because what have we done so far? So in the organic chemistry, you would have already looked at DNA being a polymer, a double helix polymer. So I just wanted to sort of remind you how all of these things link together. So a gene is a section of DNA, and DNA is a molecule made of monomers. And here is an example of that monomer. There are 4 different types of monomer, A, T, C, and G. And that's the monomer there. Nucleotide. So just reminding you of all that C10 stuff we did. We will do way more on DNA and the discovery of DNA specifically in the biology paper 1 side. I just found this on the internet and I thought it was interesting. So this is just to show you that Mendel looked into inheritance in 1865, and this was way before we are seeing or knowing anything about what the shape of a DNA molecule looks like. So it's in 1953 we proposed the actual shape, and it's not until the 90s that we're sequencing people's DNA. And I've just written at the top there, origin 1859. That's because that's when the Origin of Species was released. And so Mendel didn't had some knowledge of Darwin's work, but Darwin wouldn't have known anything about the idea of inheritance. But they sort of work together quite hand in hand about these dominant characteristics. So key things to take away from this is Mendel came up with the idea of inheritance and the idea that you have dominant recessive genes. So he bred pure white plants with pure red pea plants. The reason why his work was good because he had a large sample. And however, his fertilization was quite random. He got some theoretical ratios. He could identify his anomalies quite well. So his work was generally quite good and quite well respected. It was mostly observation based. His ideas weren't accepted straight away because we didn't know about chromosomes, genes, DNA. We didn't know that they occurred in pairs. We all had like preconceived theories about how inheritance worked and happened. Like people had noticed that you look like your father or whatever, but they didn't have a specific idea. Mendel was also a monk, so he wasn't an academic, or he wasn't from an academic establishment, so he probably would have had less kudos than other people. He also didn't put it in one of the major journals, so other scientists were unable to peer review it, and so people didn't know about it for a really long time, and so his ideas weren't corroborated until much later. If you still have a QR reader on your phone, or you can download a QR reader on your phone, we're going to do more about the discovery of DNA at a later time, but if you want to check how your knowledge is developing, secure, whether you're extending or you have mastery and are able to do the 6-mark questions, these are QR codes that link to Word documents that take you through all of the key information. So you can go through these checkpoint tasks if you wish in your own time and have a go. I've just downloaded them, check they work. I just downloaded a QR reader, check it works, and it does. It's really good. So yeah, give it a go.